My name is Kavita and today we are going to understand concept of metazoa. In this session, the objectives of the session are you are going to understand the concept of metazoa and also how metazoans have evolved. By the end of the session, you will be familiarized with the concept of metazoa. Now let's see what are these metazoans. The metazoans are multicellular organisms. You are familiar with unicellular and multicellular. If not, let me tell you. Unicellular organisms are organisms that are formed of only one cell. Multicellular organisms are multi means many, cellular of cells. So multicellular means that which is formed of many cells. So these metazoans are multicellular animals, zoon, animal, protozoon. So there again, they are the primitive animals formed of only one cell. When many cells are present in an organism, in an animal, underline animal, they are called metazoans. And the question comes, then what about the multicellular plants? They are called as metaphyta. Okay, so coming back, metazoans are multicellular animals and these are mitochondrial eukaryotes. Simply we can say eukaryotes. Now what are eukaryotes? Though all those organisms which have true nucleus, means where the nucleus is surrounded by nuclear membrane plus cell membrane bound organelles are present inside the cell. So such organisms are called as eukaryotes and all the metazoans are eukaryotes with their own mitochondria. And these metazoans are also, they have, most of the higher metazoans at least, have tissues, definite tissues, and tissues have evolved into or clustered together to form organs and then the organ systems. These metazoans have evolved from protists, so the single celled organisms, almost around 700 million years ago. Okay, now let's see the definition and the features of metazoans. So these metazoans can be defined as the holozoic multicellular organisms which develop from embryo. What is holozoic? Now this holozoic refers to type of nutrition. In organisms, there are two types of nutrition. One is autotrophic, the second one is heterotrophic type of nutrition. In autotrophic type of nutrition, auto, self, okay. So, organism can produce its own food by using sunlight or any other source of energy. For example, plants. Whereas animals, they come under heterotrophic types where they cannot produce their own food, instead depend on other organisms for their food. Now, under heterotrophism, there is one type, that is our holozoic type of nutrition. Now, this holozoic type of nutrition means here the organism will ingest, then digest and ingest the food. Okay, so if an organism ingest, digest and ingest the food, such type of nutrition is called holozoic. For example, our own kind. We take the food, we digest it and ingest it. We do not produce our own food. I am not talking about cooking, but the actual food. So, holozoic type of nutrition is where it's a type of heterotrophic type of nutrition where the organism depends on other organism for its food. Okay, so metazoans are holozoic type of organisms and they are multicellular. So, if an organism is multicellular, that's a basic one, plus it is holozoic, and then if it can develop from an embryo, okay, then it is a metazoic organism or metazoa. Okay, now here these metazoans are differentiated into usually the cells. Okay, they are first differentiated into tissues and the tissues into organs, then the organs into organ systems. There are two types of metazoans, lower metazoa and higher metazoa. This all depends upon complexity of the body. See, we are talking about evolution of a multicellular organism. All of a sudden a highly complex metazoan cannot be developed. So it is a evolution, it is a slow and step by step process. That means there are certain metazoans which have primitive features and some which have highly evolved features. That means all these lower features have accumulated and they've given rise to a highly complex form of organism. So here we have two types of metazoans, lower metazoa and higher metazoa. Just recall, you are familiar with all different phyla of animals. So protozoans, okay, then Porifera, Coelenterata, okay, then Platyhelminthes, Aschelminthes. So Porifera to Aschelminthes, they are considered as lower metazoans, where all different types of evolution is taking place, complexity is increasing, but yet they are simple. And Annelida onwards, okay, all other higher multicellular organisms, animals, are called as higher metazoans. Why this separation? It's because of difference in the complexities, because what is true for higher metazoa, all of them is not true for lower metazoans, because in these metazoans such features have not yet evolved. Okay, hope I have made myself clear on that particular thing. Now let's see other features of metazoans. So metazoans show symmetry, okay, or they might be asymmetrical. Now what is the symmetry? Symmetry is, it's actually the arrangement of body parts in one particular geometric pattern. I'm sure you have studied symmetry in chemistry, molecular symmetry, okay, or maybe in lower classes the symmetry of some objects. Okay, now here we are talking about animals. So animals also show symmetry. What is symmetry? It is the arrangement of different body parts in a geometric pattern. To make it very simple, so it is the arrangement of different body parts, okay, around a central axis. How these different parts of the body are arranged around a central axis in the body is called as symmetry. If there is no such pattern, okay, no such order in the arrangement of the organs around a central axis, okay, then such condition is called asymmetric. So what you are seeing here, okay, there are two types. One is asymmetry. Asymmetry means there is no proper pattern. That means the different structures of the body are not organized in a particular pattern around a central axis, as you can see in amoeba. So where I can draw a central axis, it's very difficult. And let me say that if I draw a central axis here, on both the sides of the central axis there is no proper pattern, they are unequally distributed. Same is true here too, unequal distribution. Whereas in other groups, in higher groups, okay, the organs, different organs of the body are arranged in a particular geometric pattern. So such a pattern is called as symmetry. Okay, and in symmetry there are different types. We are talking about symmetry in case of metazoans. So in metazoan we have radial symmetry, bilateral symmetry and biradial symmetry. Whereas in protozoa you will also come across. So if you are talking about only animals as a whole, then there is also spherical symmetry. So now here I'll only discuss on three types of symmetry: radial, bilateral and biradial. So what is a radial symmetry? A radial symmetry is the one where the different parts of the body are arranged around the central axis in such a way that, okay, so if a section passes through the central axis, it can give you two identical halves. So if the body can be cut into identical half in any plane passing through the central axis anterior posteriorly, then it is called as a radial symmetry. Now you see here we have a picture of a hydra and I am drawing a central line here. Excuse me for that unequal line, but just imaginary one. Now you see, if it passes through the central axis, okay, in any planes passing through the central axis, for example this one, if this is one plane, you are going to get identical half. Okay, next if there is another, okay, this one, in this plane you are going to get identical half, in this plane you are going to get identical, in this plane identical half. That means in all different planes which passes through the central axis anterior posteriorly, you are going to get identical half. I'm saying anterior posteriorly. Why not all the length? Because if I cut it here, I won't get identical half, isn't it? So anterior to posterior, I'm going to get identical half. So this kind of symmetry is called as radial symmetry. Okay, in coelenterata you find radial symmetry. Whereas if it has to be spherical symmetry, then in all planes, all different planes, if you get identical half, then it is a spherical symmetry. In metazoa you find radial symmetry. Now what is a bilateral symmetry? In this case, only in one plane, okay, only in one plane that passes through the central axis, you are going to get identical half. For example, this is a butterfly. So if I cut exactly at the center, okay, exactly at the center, here you will be able to see the wrong diagram lines. Okay, just give me a second. I'll draw the line here. Yes, so this one. See here, you are going to get identical. This is one half, this is one half. Identical half. In this plane you won't get. In this plane you won't get. In this plane also you won't get. So in only one plane passing through the central axis, if you could get identical half, then it is bilateral symmetry. And then we have a next type, biradial symmetry. It is a modified radial symmetry. See, in case of radial symmetry, in all possible planes, okay, in all possible planes, you will get identical halves passing through the central axis. Whereas in biradial symmetry, it's a fusion of bilateral and radial. Only in limited planes you are going to get identical half. So then it is called as a biradial symmetry. Okay, so in metazoans show radial, bilateral and biradial symmetry. Metazoans also show cephalization. Now what is cephalization? Cephalo comes from kephale, means head. Okay, so formation of head is seen. Again, it is not true for all the metazoans. So cephalization is seen in case of higher metazoans. Formation of a distinct head. The anterior portion, which is considered as a distinct head, okay, is seen in higher metazoans, whereas in lower metazoans, cephalization is lacking. The next feature is coelom. Coelom means a body cavity. Coelom refers to cavity. So here there are three conditions. One is acoelom. A means without, coelom means body cavity. So there is no body cavity. That means the spaces between the different tissues, okay, are, yes, now if I just focus it on there, what you are seeing here is a acoelomate. That means, see, there are organs, there are different organs, and the space between these organs and the body wall is filled with the parenchymatous tissue. There is no space left between these two. So this condition is called as acoelom, without body cavity. In case of platyhelminthes, you find this condition acoelom. Then there is a pseudocoelom. Pseudocoelom means, pseudo means false. False body cavity. Now what is this false body cavity? A body cavity, okay, there is a cavity, okay, but it is false. Now why it is false? Because this body cavity is not derived by the mesodermal splitting or outpouching of the gut. Okay, so instead simply it is a space that is produced at the time of embryo development. Okay, so that's why this cavity, which is present in case of Aschelminthes, as you can see here, is called a pseudocoelom. It usually develops from a blastocele. Okay, the next one is eucoelom, true coelom. Okay, U means true, coelom, body cavity. Now, what is a true body cavity? A true body cavity is the one where the body cavity is formed either by the splitting or outpouching of the gut, okay, and here it is lined by epithelial cells. You see here the both, see, endodermis is there, then you are going to see the intestine, and around those organs there is a layer of epithelial cells. Okay, a layer of epithelial cells which are usually derived from the mesoderm, whereas such epithelial layer is absent in case of pseudocoelom. Okay, pseudocoelom doesn't contain a mesothelium or the mesodermal epithelial cells. In case of eucoelom, the body cavity lined by epithelium internally and externally is called as a true coelom. So this true coelom is seen after Aschelminthis. That means from Annelida onwards, you are going to see the true coelom. Again, in true coelom, there are two types of true coelom. One is schizocele, the other one is enterocele. So schizocele is formed by the splitting of the mesoderm, splitting of the mesoderm, whereas enterocele is produced by the outpouching of the gut. The gut will form small pouches, then extends in both the direction, continuing forming a cavity, okay, between the internal organs and the body wall. So there are three conditions. One is acoelom, no body cavity at all. The space between different organs is filled with the parenchymatous tissue. Next one is a pseudocoelom, where the body cavity is not lined by epithelial cells. Okay, and the third type is eucoelom or the true coelom, where the body cavity is present and it is lined by epithelial cells and derived from either mesodermal splitting or by the outpouching of the gut. Okay, so the next feature that we can see in case of metazoans is the metamerism. Now, what is metamerism? Metamerism is a longitudinal division of the body into linear series of similar sections. So there are certain things. It is a longitudinal division. Okay, longitudinal division of the body into linear series. Linear means passing in a single line. Okay, similar sections. See, you see here. Okay, here. Yes. See, these similar sections. This section and the first section and the second section. This one and this one. They are similar. As the term here itself says, you see. It says they are similar sections or parts. How they are present? They are present linearly, one after the other. And it is a longitudinal section, division. That means along the length of the body, okay, if there is a repetition of body parts in a single line, then it is called as a metamerism or segmentation. There are different types, okay. True segmentation and false segmentation are true. False segmentation is seen in case of tapeworms, whereas the true segmentation is seen in case of Annelida onwards. Aschelminthis, up to Aschelminthis, there is no metamerism. So there is again exception, you see. Lower metazoans up to Aschelminthis, we consider them as lower metazoans. So up to Aschelminthis, you won't find metamerism. Annelida onwards, you are finding metamerism. Again, there are exceptions. Anyway, but still, generalized rule is that Annelida onwards, they show metamerism, or at least normal segmentation is seen. So segmentation, metamerism, is one of the feature of so-called higher metazoans. Okay. Next, there are different theories how actually metazoans have evolved. So these metazoans have evolved, but maybe, as we said, from the protozoans. Next question is, how exactly from protozoan? Is it bundling of protozoan cells and then differentiating, or a single protozoan itself gave rise to a multicellular organism? We don't know. So there are different theories that explains how exactly these metazoans formed. We are going to see one by one. At least we will discuss 3 to 4 such theories that will help us to understand how the metazoans are formed. The first one is syncytial ciliate theory. Syncytium, you must be familiar. If not, let me tell you. Syncytium is a condition where a single cell has many nuclei. Syncytium. Cenocytic condition is another term used for that. So, where a single cell will have many nuclei. Now, here this theory says that metazoans have evolved from ciliates. What are ciliates? These are unicellular organisms which have cilia as their locomotive structures. So, a syncytial ciliate has given rise to a metazoan is what theory says. Okay, and this theory was first actually proposed by Hudzi in 1953 and 1963, then supported by many other scientists. So, it says that metazoans, the present multicellular organisms, have evolved by the nuclear division. You see here, this is a syncytial condition where there are many nuclei within a cell, and it is a ciliated protozoan it is. After these nuclear divisions, okay, each nucleus was surrounded by its own cytoplasmic membrane, resulting in something called as cellularization. So, when cellularization has occurred, this cell is no longer a unicellular one, it has become a multicellular one. Okay, so it has become multicellular. This is how metazoans are formed. This is one view, one theory. The next one is colonial flagellate theory. So this was proposed by German scientist Haeckel and then supported by Lancaster and others. So as you can see here, it is a flagellate theory. That was a ciliate one. So they thought that ciliate protozoans have given rise to metazoans. This they say that flagellates have given rise to the metazoans. Colonial, Volvox colony. Volvox colony. It is a colony of protozoans, isn't it? So many flagellated cells form one bundle or multiple bundles rather. So such colonial flagellates, okay, which were actually once independent cells were living in a colony, have become dependent on each other, then started distributing the work, division of labor, and slowly some groups of cells have evolved into different types. Okay, and that has given rise to the metazoan concept or metazoan status. That's what colonial flagellate theory focuses on. As you can see here, there is a colony. Okay, and then what happens? These colonies, then they slowly, so here they were all independent ones, and then slowly what happens? Differentiation takes place. Some of them are flagellated, some of them have lost the flagella, and then they become dependent on other cells, and then they started distributing their own work, and then slowly modification has taken place. This is what the theory says. The next one is colonial blastia and planula theory. And as per this, okay, here it focuses on, like the colonial flagellate theory, here they say that okay, there were flagellated colonial protozoans, okay, and at the center there was a hollow space, and then what happened? Slowly this structure that was formed of outer layer of flagellated cell center and the cells, and then the center hollow, that was similar to the present blastula concept. Okay, so it is called as a blastia. And then what happens? Gastrulation takes place where the invagination of the cells occurs. Na? So similarly, invagination has resulted in formation of a gastria or a planula. Okay, and then slowly later on it has given to the higher groups. Okay, so that is the concept of colonial blastia and the planula theory. And there are certain evidences like sperm cells are flagellated in all the metazoans, and in the lower metazoans they have like monociliated cells within the body. For example, in sponges you find collar cells. Okay, so that can be considered as a evidence. The next one, the most recent one rather, polyphyletic origin of metazoans. So Greenberg and his followers do not believe that only one phylum has given rise to metazoan. So they believe that the present day metazoans have evolved from different phyla. That's what polyphyletic. Okay, single one group it hasn't given rise to the new forms. Instead, as you can see in the picture, so some of the flagellates okay, have given rise to sponges, the anidarians, the present day. Whereas ciliates have given rise to ctenophores, and from their acellules and then from their other metazoans have evolved. That is what polyphyletic origin of metazoa focuses on. Okay, so that is about the metazoan concept. Now let's test your understanding of the topic. So first question is, syncytial theory explains. First option, formation of xylem. Does it explain the symmetry of animal, origin of life, origin of metazoa? So these are the four options you have. So does it explain formation of xylem? Does it explain the symmetry of animal, origin of life, or origin of metazoa? Hope you have your answer ready, and the right answer is D. It explains the origin of metazoa. The next question is, syncytial theory was proposed by. First option is Hansen and Hetze. Second is L.H. Hyman. Third is Lancaster. The fourth one is E.H. Haeckel. Now, just think your answer. Hope you have your answer, and the answer is A. Syncytial theory was proposed by Hansen and Hetze. Your third question is, on the basis of body organization evolution, animals are grouped as parazoa, metazoa. Body organization evolution, read that once again. Protozoa, metazoa. Protozoa, parazoa. Metazoa, eumetazoa. Okay, so these are the options. Read the question again. On the basis of body organization evolution, animals are grouped as. Hope you got the answer, and the answer is B. Protozoa, metazoa. Protozoa, single-celled, metazoa, multicelled. Next one is unique feature of metazoa are. What are the unique features of metazoa? Multicellular, extracellular matrix, interdependent cells, all the above. Is it multicellular feature? Is it extracellular matrix present, or interdependent cells, or all the above? The answer is D, all the above. The last question, which group of protozoa support and give best explanation of colonial theory of origin of metazoa? Colonial theory of metazoa, okay. Ciliata, Sarcodina, Flagellata, Sporozoa. Ciliata, Sarcodina, okay, Flagellata or Sporozoa. Among these four, which one is the correct answer? The protozoa that supports colonial theory of origin of metazoa. And the answer is C, Flagellata. Colonial Flagellata theory, remember? So that is the right answer. The summary of the session are, metazoan animals are multicellular, mitochondrial eukaryotes they are, and they are covered by, each cell is covered by cell membrane, and their cells are interdependent, and gonads present, okay, except lower metazoans. They show coelom, symmetry, metamerism, etc., etc. That's all for today's class, and the references for my today's class are these. Thank you.